Okay, I'd like to go back and talk now about protein secondary structure. And when we say secondary structure, we're talking about alpha helices and beta sheets. But just to tie this back, remember when we folded the tuber into this complex 3D shape following basic principles of chemistry. So we went from the primary sequence, the primary structure of a protein, which is just the linear sequence of amino acids, to what's known as the tertiary structure of the protein, the folded protein structure. And we skipped secondary structure. Where are the alpha helices and beta sheets? They're not in this folded protein. So that's what we want to drop back and discuss. Because in a real protein, like this protein here, you have combinations of red alpha helices and yellow beta sheets. So how do those things form? So the important thing to remember about secondary structure is maybe two things. The alpha helices and the beta sheets are very consistent structures, they're very stable structures, and they provide sort of a rigid platform or scaffold upon which the protein is built. The second, so they're consistent, they're stable, they're stabilized by hydrogen bonding that goes from backbone atoms in one strand of a beta sheet to backbone atoms in another strand. Or in an alpha helix, the hydrogen bonds go from the carbonyl oxygen of one amino acid to an amino nitrogen of another amino acid that's 4 residues further along on the helix. So that's what you want to focus on. And what I have here is another model then of either an alpha helix here or a beta sheet here. And the model starts with these little backbone units of the amino acids. And I'm going to try to put these, set these next to each other so that the nitrogens are all out here. But look how different the oxygens are. So these are both amino acids, and they're interconvertible one from the other, even though you can't rotate these. But remember when we were talking about this model of a protein, we talked about how you simply rotate around these single bonds, and that changes the shape of the protein. So imagine you wanted to coil this up into an alpha helix or to a beta sheet. What you would do is you'd have to set the rotation around 2 of these angles such that it would give you a consistent alpha helix or beta sheet. So in these units, they've been set that way. So you take one amino acid and you make a peptide bond with the next amino acid. So we have magnets here that allow you to form these peptide bonds very quickly. And when you do that with this amino acid unit, what you notice is that it creates this right-handed alpha helix. How do you know this is right-handed? It's right-handed because you imagine it's a spiral staircase, and as you walk up this spiral staircase, it'll be your right hand that will be on the outside rail of the staircase. The other thing you should notice is that when I hold it like this, all these red oxygens are pointing up, and they're pointing toward blue nitrogens. Okay, if you were holding this in your hand, well, you can actually see right now how unstable this helix is. All right, watch what happens when I take a hydrogen. The hydrogen initially starts off as being associated with the nitrogen, but when this slightly electronegative oxygen comes into close proximity, it shares that hydrogen in a hydrogen bond. And as you snap these hydrogens, as you form these internal hydrogen bonds in an alpha helix, it immediately contributes lots of stiffness to this helix. So what started off as a very floppy helix with those hydrogen bonds now becomes a very stable structural member of a protein. Okay, so hydrogen bonding between carbonyl oxygens and amino nitrogens stabilize alpha helix. In the same way, if you put these backbone units together, they have angles set in them so that they create this extended zigzagged pattern. So I've got two of them here. And notice that in this one, we've got the blue nitrogen here, we've got a red oxygen, so it's the amino terminus here, the carboxy terminus here. And we can arrange the other strand of this beta sheet in the same direction, in which case this would be a parallel beta sheet. Or we can turn this one around. Now the N terminus is here versus here. So this is going to make an anti-parallel beta sheet. Both kinds of beta sheets occur in proteins. That might be something you want to pay attention to as you're looking at your protein. The hydrogen is initially associated with the nitrogens, and they are going to hydrogen bond with oxygens on the other strand. So I've run out of, let me steal one from here. So there's the hydrogen bonding then that stabilizes one strand of an anti-parallel beta sheet with respect to the other. So I have in front of me here three different proteins. This is an opioid receptor, which is made up almost entirely of alpha helices. These are transmembrane alpha helices. All of them are internally hydrogen bonded like this, and they give this protein a very distinctive stable shape. Here's a protein, the green fluorescent protein, which is sort of the opposite in that it's made up almost entirely of beta sheets. This is actually known as a beta barrel. I think there are 11 strands of protein that make up this beta barrel. Again, all hydrogen bonded across from one strand to another. Makes a very stable structure for this protein. And then here's a protein. This is the Sepia Terian reductase protein. And it's sort of more common in that it has both secondary structural elements in it. It has red alpha helices. It has a very nice 7-stranded beta sheet shown here in yellow. That provides the basic scaffolding upon which active sites can be created, usually in the loops that connect one secondary structural element to another.